Hi, everyone. Up to this point, we've mostly focused on structures inside the cell, like the plasma membrane and the cell wall. But many prokaryotes also possess structures that extend outward from the cell surface. These external structures allow microbes to move, attach to surfaces, communicate, exchange genetic material, and interact with their environment in complex ways. Some help cells swim towards nutrients. Others help microbes colonize surfaces and form biofilms. Some even contribute to pathogenicity and disease. In this lecture, we'll explore the major external structures of prokaryotic cells and how they influence survival and environmental interactions. By the end of this module, you should be able to identify and describe the function of major external structures of prokaryotic cells and explain how external structures contribute to movement, attachment, and environmental interactions. Many prokaryotes are capable of movement, and one of the most important structures involved in motility is the flagellum. Flagella are long protein appendages that rotate like microscopic propellers, allowing cells to move through liquids and across surfaces. Different bacteria can have different flagellar arrangements. Some bacteria have a single flagellum at one end called a monotrichous arrangement. Others may have a tuft of flagella at one pole called lophotrichous. Amphitrichous bacteria possess flagella at both ends of the cell. And some bacteria have flagella distributed across the entire cell surface, and they're called paratrichous flagella. These arrangements can slightly influence how cells move through their environment. For example, paratrichous bacteria often alternate between coordinated runs and tumbling movements, while polar flagella may support smoother directional swimming. Motility is extremely important because it allows microbes to move towards favorable conditions, such as nutrients, oxygen, or light, and allows them to move away from harmful environments. In aquatic and soil environments, especially, movement can strongly influence microbial survival and ecological success. Movement alone is not enough for survival. Microbes also need the ability to sense changes in their environment and respond to them. Many microorganisms can detect environmental signals such as chemicals, oxygen levels, or light. Chemotaxis refers to movement in response to chemicals. Cells may move towards nutrients or away from toxic compounds. Some microbes also perform phototaxis, which is movement in response to light. As you look at the image on this slide, notice that microbial movement is not random. Cells can bias their movement towards favorable conditions and away from harmful environments. These responses help microorganisms locate resources, avoid stress, and survive changing environmental conditions. Many prokaryotes also possess external attachment structures called pili or fimbriae. These are short protein-based appendages that extend from the cell surface. Some pili help cells attach to surfaces, tissues, or other cells. This attachment is extremely important for colonization and biofilm formation. As you look at the image on this slide, you can see a specialized pilus called an F pilus or fertility pilus connecting two bacterial cells. This structure is involved in conjugation, a process where bacteria transfer DNA directly from one cell to another. Through conjugation, bacteria can rapidly spread useful genes, including antibiotic resistance genes, through a population. And additionally, some pili and fimbriae can be used for another type of movement where you can move across a surface instead of like the swimming flagellar movement. So while some pili primarily function in attachment, you have these others that can play important roles in communication, exchange of genetic information, and movement between environments. As you look at the capsule stain on this slide, notice the darkly stained bacterial cell surrounded by the lighter, unstained regions around them. Those clear halos are the glycocalyx. The glycocalyx is an external coating that surrounds some prokaryotic cells. In capsule stains, the cell and background are stained, but the glycocalyx itself often does not take up the stain well, allowing it to appear as a clear region around the cell. There are two major forms of glycocalyx. Capsules are highly organized and firmly attached to the cell surface, while slime layers are more loosely associated and diffuse. These structures provide several important advantages for microbes. They can help cells attach to surfaces, retain moisture, and protect against drying. In pathogenic bacteria, capsules can also help cells avoid immune defenses by making it more difficult for immune cells to recognize or engulf them. So, although the glycocalyx may appear as just a faint halo under the microscope, it can have major effects on microbial survival and pathogenicity. Many microorganisms do not live as isolated individual cells. Instead, they often live together in organized surface associated communities called biofilms. As you look across the diagram on this slide, you can see the general progression of biofilm formation. In the first stage, free swimming bacterial cells attach to a surface. Over time, those cells begin to divide, recruit additional cells, and produce an extracellular matrix around themselves. That matrix is the light-colored material surrounding the cells in the image. It is made up of substances such as polysaccharides, proteins, and DNA, and it helps hold the community together. As the biofilm matures, the structure becomes thicker and more complex. Channels can even form within the biofilm that allow water and nutrients to move through the community. Eventually, some cells may leave the biofilm and disperse to colonize new environments. Biofilms are extremely common in nature. They form on rocks and streams, plant roots, soil particles. They even form on medical devices and even human teeth as dental plaque. One reason biofilms are medically important is that cells within a biofilm are often more resistant to antibiotics and disinfectants. The extracellular matrix can help protect cells, and the close association of microbes can change how they grow and respond to stress. So rather than thinking of microbes as always living alone, it is often more accurate to think of them as members of complex microbial communities. In this lecture, we explored several important external structures found in prokaryotic cells and how those structures influence survival. Flagella allow cells to move through their environment and respond to changing conditions. Pili and fimbriae help microbes attach to surfaces, interact with other cells, and even exchange genetic information. The glycocalyx and biofilms provide protection, help retain moisture, and support the formation of complex microbial communities. Together, these structures allow microorganisms to interact dynamically with their environment rather than simply existing as passive cells. But even with these adaptations, many environments can become so extreme that normal cellular activity cannot continue. Nutrients may disappear, temperatures may rise dramatically, conditions may become too dry or chemically stressful for active growth. Some bacteria have evolved an extraordinary survival strategy for these situations, the ability to form endospores. Pardon me. In the next lecture, we'll explore how endospores form, why they are so resistant, and how they allow certain bacteria to survive some of the harshest conditions on Earth. With that, thanks so much for attending this lecture, and I'll see you next time.